And the bigger the horn, the more bass you get. The smaller the horn, the more treble you get. Well, look at the architecture of the cochlea. Different areas are more sensitive to different notes. But that's not the only way we can separate notes in the environment. Some people are really, really good at it, like a conductor. He can be conducting his entire orchestra, and then they'll say during practice, stop, stop, and he'll point to one person and address one note that that person's playing and say, that was slightly out of pitch, you need to play this note, not the note you're playing. Out of all of that noise everybody is making, this one person can pick out one note coming from one person in the orchestra. Wow, that's phenomenal. And some people can do that. Not everybody can. Sorry. We wish we were all that lucky. Because their flavor, their sound that they perceive is so much more enriched than most of us perceive. Okay? So yeah, the organ of Corti would be right here. And here it is. An artistic rendition of the organ of Corti. So here is the cross section, okay? Here's your cross section right here, and here it is magnified in this picture. Your organ of Corti is right here. And here's your scala vestibule, scala tympani. Those are the two canals. And the scala media is in between. Media, middle, in between the vestibule. These two are connected. This one's not. So you have a basilar membrane right here. Here's your basilar membrane. And you have a tectoral membrane sitting on top. Here's your outer hair cells. This is your inner hair cells. It's your inner hair cells that here, not your outer hair cells. Your outer hair cells. See this tectoral membrane, it vibrates. Okay? And the outer hair cells adjust the tension of the tectoral membrane. So you can adjust the frequency that can be heard by adjusting the tension of the tectoral membrane, which will in turn alter the vibration frequencies that the inner hair cells can perceive. Wow. So we can adjust to notes. So this is the other way in which we could perceive different notes, in addition to the architecture of the cochlea itself. Okay, so I thought I'd take pictures of this too. Okay, here's your helicotrema. Here's your vestibule media. Okay, the top would be the scala vestibule, the bottom would be scala tympani. Okay, this is your bony lambret. So all this, and look, if you look close, you can see perversion systems in your bone. This is in your head. Okay, this is your skull. So it comes this way and then back out. And each one of these would be the organ of Corti. So here is your helicotrema, and I don't know what the heck that is in the helicotrema. Here is your organ of Corti right here. So let's check it out. Let's see. Here's your outer hair cells. Here's your inner hair cells. Here is the tectoral membrane. Okay. Okay, and here it is at higher power. You can see the inner hair cell there. And it's not hair at all. It's cilia. It's not hair. Okay? And nobody seems to want to change the name. See, there they are, the little cilia right there. And this one right here, the cilia looks a little thick for some reason.
What's such long fruit?
Artifact. Something came off, and if you notice, it's in several places because this is a montage of several images stacked on each other. That thing is not even supposed to be there. Okay. I do have the equipment to erase it, rid of it, but I said, yeah. You know, keep in mind, this is a montage. Several images stuck together. It took me several hours to generate this image. It was anywhere from 15 to 20 minutes an image that I spent on this. Okay. Here it is under higher power again. Even higher. Now it really looks cool. Okay, here's the tectoral membrane, vascular membrane, inner and outer hair cells. Okay, and these right here make solution. Okay, these just make some of the solution in change. Okay, so here's the way it works. The hair cell is really a kinocilia attached by stereocilia. So when you bend it, it will either fire an inhibitory information or excitatory information. And these are hair cells. They're epithelia cells that make a cilia, a stereocilia. And these stereocilia, if you can see, they're connected to each other. So when you bend one, you bend them all. Okay? And they're mechanoreceptors. You bend them, they fire action potentials. And they stimulate the release of neurotransmitters on the primary afferents. Okay? The neurotransmitter is glutamate. Okay? So, who cares? So here's some electron micrographs of the same thing. You can see the connections between the stereocilia. Here's the stereocilia, kinocilia, and the kinocilia and the stereocilia. You can't see it in the stereocilia as well, but it's a 9+2 doublet. That's a cilia, not a hair cell. The 9+2s, guess what that is? Biological motor, microtubules, and those little kinesins and dyneins. That's the 9+2 doublet. So I don't really care if you even understand this or not. So I'm not going to spend a lot of time here. The bottom line is you bend the cilia and you fire an action potential. If you want the detail, you can be my guest. Try and figure it out. I wrote all this in here. Okay, so this is my wording. And I read it the other day and I said to myself, what in the hell are you trying to say? Okay, so I mean it made sense when I figured it out and wrote it, but now it would be just like I would be doing just what you would do, looking at this, trying to figure it out. So, and I wrote this a really long time. Again, before I had a job. In between jobs, actually. So here it is again, nothing new, okay? And again, still nothing new. Here's the tectoral membrane sitting on top. Okay, so you bend it and you can reshape it, and as a result you can change the position of the tectoral membrane and sense different frequencies. You can alter the frequencies to hear what you want to hear, in fact. And that's how conductors can sort out one note in a crowd, because they pitch to it. Okay? So the pathways for hearing, okay? Spiral ganglion from the hair cells, the spiral ganglion, to the cochlea nuclei and the medulla. Then they cross over to the contralateral side, ascend. Some of them cross back, and then they go to the thalamus, and then they're relayed to the geniculate nucleus of thalamus, and then they're relayed to the cortex, auditory cortex. Conduction deafness sometimes hampers sound conduction, the fluid of the inner ear. Okay, conduction could be just too much earwax in your ears, or a perforated eardrum. Anybody ever perforate your eardrum? Just me. Nobody in here? I blew my eardrum out on this side. Whatever you do, if you ever blow your eardrum out, don't put water in your ears. It will hurt as like you would not believe. It's worse than passing a kidney stone. Okay? If you get water in your ears when you perforate your eardrum. Huh? You don't. Yes. Okay. I don't want to sit here and talk about it. It's sad. Okay, so ossical sclerosis, in other words, arthritis of your middle ear bones. You lose. That's the person that's sitting in the car with a boom, 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 right? His radio going really loud. They're going to have ossical sclerosis. Okay? They're going to become less able to hear. Sensory neural deafness results from damage of the neural structures at any point from the cochlea hair cell to the auditory cortex. Tinnitus, ringing in the ears, right? Anybody in here have tinnitus? Just me?
No.
You have? I have tinnitus, and I can really hear it on this side. It's only in this ear, and it's always wee, really, really high pitched. It's going on all the time. And I just ignore it most of the time. Now that I'm not ignoring it, I can really hear it loud. I can tell when I'm going to get sick. Because when I get starting to get sick, it gets louder. I say, hey, you're going to get sick. Okay? And it does every time. Meniere syndrome, Lambert disorders that affect the cochlea and the semicircular canals, called vertigo, nausea, and vomiting. Okay, so those are the anomalies. So now we're going to switch to the vestibular apparatus for equilibrium. Here's the vestibule, the sacula is in it, and the utriculus is in it. Okay? The utriculus is attached to the semicircular canals, and the sacculus is attached to the cochlea. Okay? But they're both involved in maintaining balance and equilibrium. Okay? And they do it with hair cells just like hearing. It's the same type of hair cells, just designed to do a different thing. And here they are. Here's the hair cells, and they're sitting in this otolith, otolithic membrane. And it's really not a membrane. And then you have otoliths, which are calcium carbonate sitting on top. Okay, now visualize this. You make a bowl of Jell-O, right? And it's a dome of Jell-O. Okay? You take the Jell-O out, and you got this bowl, and you shake it, right? It vibrates. That's what the otolithic membrane is doing. Vibrate. Okay? Yeah, it's just like Jell-O. Now, if you stuck some rocks on top of the dome of Jell-O and you vibrate it, it will vibrate more vigorously. That's why you have these little calcium carbonate crystals sitting on top of the otolithic membrane, which is just a gelatin made out of glucosamine, glycans, gangs. Right. So that's how you get the vibration and the bending of these cilia and stereocilia. So they're in the macula of the utricle, and then they're in the sacula as well. And that's for just standing here being balanced. Okay? Now you have this same type of apparatus in the semicircular canals. Okay? So in the semicircular canals, well, we're not going to do that. So what do they sense? They sense static equilibrium. They also contribute to some dynamic equilibrium. Okay? So, and here's what they look like again. Nothing new here. Okay? So, and the semicircular canals, as well as the utricle vestibule, or the vestibule which contains the sacula and the utriculus, when you move, they bend the hair cells. So the semicircular canals are in different directions. Remember what they look like? They were in different directions. That allows you to sense movement in different directions. You have a three-dimensional perception of the environment when it comes to movement because of the architecture of the semicircular canals pointing are circular in a different plane. And when you move, one semicircular canal may bend hair cells more than another depending upon the plane that semicircular canal is in, and you decipher that information and determine in your brain which way you move. Okay? Which is really, really weird too. Okay, three semicircular ducts or canals, okay? They allow you to sense dynamic equilibrium. In other words, equilibrium in motion. Okay? So, and here's what they look like. Okay? In the semicircular canal. And like I said, each canal is in a different plane. So you can sense information in different planes of movement at all times. Now, if you had an ear infection, everybody in here have an ear infection?
Yeah.
And you're okay if you don't move, right? If you don't move, you're okay. But if you move, what happens? The whole room starts spinning, right? How about that night you went out and you got filthy drunk? I know what
I don't drink.
Okay. You did. You did. And we all have. Some of us don't learn our lesson. We do it 3 or 4 times before we figured out, I better not do that again. I'm so sick. Right? And the room is spinning, right? Well, the alcohol content in you is so high, it's seeping into this fluid and it causes movement of these hair cells. Drunk, right? Okay. I've done it too. Okay. So if you have bacterial infection in there, bacteria don't sit around. They move around. They have cilia. And when they start moving these hair cells too. And all of a sudden the room is spinning. Okay?
When kids get tubes in their ears,
Pardon me?
Kids get tubes in their ears.
This is through the tympanic membrane, not through this. Okay? Different. Okay? So basically, that's all this is saying. The movement, you bend the hair cells one way, you'll fire action potentials. You bend the hair cells another way, you'll be inhibiting, or hyperpolarizing one way, depolarizing another way. And your brain sorts all that information out dependent upon which hair cells are moving the most, dependent upon which plane they're in within a particular semicircular canal. And you process all that information that's coming. And you figure out what's going on. So here it is. Low power. So again, 15 to 20 minutes an image. 1, 2, 3, 4, 5, 6, 7, I'll stop. Okay, a lot of images to process this.
Wow.
Because I have to take a picture, move it, take a picture, move it, take a picture, move it, take a picture, move it. And then I make TIFF files with my system, the system I use, and I have to transfer those into JPEGs. So I have to pull them up in a different program. And then I have to print screen, send that over to the different program and transfer it into JPEG, each image. And then I have to put it together in a PowerPoint and then piece them together like a puzzle. It is a puzzle. And then if I wanted to, I could check it out.
You know what you could do? You could make puzzles for people that are learning, students that are learning this. Sell them.
Hey.
That's a good idea.
Make some money.
Okay, you can come down my house and put together the next one. Okay?
No.
I'll even supply the beer and get you filthy drunk.
I give you the idea, so there you go.
Okay, so here it is again. Okay, so this is the gelatinous material, and your hair cells are right in here. Okay, cool, huh? Those are little hair cells, the little red things.